right, in this video, we're looking at something very similar as in this, I should say, in this worksheet. We're looking at something very similar to begin with what we had in the last worksheet. We're just adding a little bit to the idea that there's now something called 3' and 5'. There's a directionality here on the DNA and the RNA. So notice that we're moving from left to right, 3' to 5', and we're using T's. So this is a single-stranded piece of DNA. Not worried about the complement yet. And we're moving 3' to 5'. Well, that's important because moving 3' to 5' is the way that the polymerases move. The polymerases always move 3' to 5'. The ribosome, on the other hand, you will find will move 5' to 3', but we won't talk about that yet. That's later. But yes, the polymerases will move like DNA polymerase and RNA polymerase, move 3' to 5'. All right, so we're moving this direction, and it says in the previous worksheet, DNA was read left to right. In reality, it has a chemical directionality. So in other words, if I were to turn this upside down and we had 5' over here and 3' over here, the polymerase would move this way. So you always move 3' to 5'. If you don't have anything, you can assume left to right. Now the complementary strand is here. All right, and you can see how this works for the double helix. The top strand, of course, starts with 3' and 5', but the bottom strand is going to go in the other direction. This strand can be read just as easily as this one, and you can even have genes going opposing directions. So you could have this sequence as part of one gene and this sequence going the other way, part of another one. The polymerase would move this way up here, down here, and the polymerase would move this way up here because the polymerases move 3 to 5. All right, now notice the stops. Let's see, anything else? Now, what is the mRNA product produced from the bottom strand? Well, the bottom strand is here, so we're reading in this direction. All right, and then what is your RNA product? Well, your RNA product, all right, would be 5'. All right, which one is 5' and prime? So the RNA is, if we're reading this one to this one, your RNA is going to be this way to this way. But you know, if you just put 5 and 3 on here, you can get it the correct direction. but it should be just like this, except for it's RNA. So you would change the T's to U's. So if we're looking at this one going this way, it would be the same as this one going this way to this way, just change the T's to the U's. If you understand that, great. You need to read it. You need to understand what's going on here in this worksheet. Doesn't have the components needed for translation, not transcription. Transcription would be a Tatabox. That's pretty much all you need. You'd need a promoter and a Tatabox to get it going, and that would be, and then a start and stop codon probably, but those are the only things you would need for transcription. But translation, what would you need? Well, translation, you need a 5' end. All right, so is there a 5' end? Yeah, well, of course, we'd have a 5' end. You're just making it gives you a 5' end. And then you need a start and stop codon. So a start and stop, you actually don't need a stop, but you do need a start. And the start is an AUG, and so if you translate this, is there anything going this direction that would give you an AUG? Well, remember, we're translating it, so that would be, if you think about it, a T, what? A TAC. So it would be a TAC going this way. And I'm just going to say, I don't know. You'd have to look on here and see if there's a TAC, all right, going this way. All right, so think of it that way. You're going to make this is the complement, so that's going to be your RNA, but it's going to go this way. So we see a TAC going this way? I see a TAC going this way. All right, but is that the direction that you read this? Remember, the ribosome is going to read this way, so I'll leave that to you to figure out. All right, that's a big hint there. Does this strand have the components needed? I'll just leave it there, and you can figure it out. All right. Now, um, this talks more about the polymerase. It talks about the Tata box. Um, and then it explains that ends are when we don't know. There are unknown sequences, and sometimes you don't know if it's an A, G, C, or T, and so you would put an N there. All right, now minuses are behind the transcription start site. So here's where you learn about the transcription start site. Not important in this class, uh, so you don't have to know particularly anything about the transcription start site, but that's going to be where, even though the polymerase binds at the promoter, it really doesn't start copying until you get to this transcription start site. We don't have to know much about that in this class, but it's good to know. All right, there is a gap there between. Just because it sticks somewhere doesn't mean that's where it's going to copy. It's going to move down a little before it starts copying. All right, I'm just going to leave that there and let you read it. Now add a functional Tata box. So a Tata goes on what end? Well, it goes in the DNA, not the RNA. So it's going to be in the beginning. You can see this in the book and the PowerPoints I gave you. The Tata box is the promoter, and a TATA is going to go at the beginning of the gene. So you're going to find it somewhere in front of the place where, like in front of the, or before, if you want to think of it that way, before the start codon. And it's going to be on the what side? Well, we would normally say to the left, but the left would be 3', the 3' side of the start codon. So that gives you an idea of where to put a Tata box. All right, and read through this. Um, a hairpin loop is a terminator. We aren't going to go into details in this class on this. If you like this stuff, contact me. I'll give you some research to work on. If you really want to learn about this, I teach a biotech class where we cover this material. Now, adding sequences to the hairpin loops for two complements such that two genes can exist. I'm going to make this piece extra credit. So if you can figure this out, you'll get extra credit. If you don't, it won't count off. All right, but this is the hairpin loop. This is the thing that makes the RNA polymerase fall off. All right, and if you can figure this out, I'll give you credit for it. So give it a shot. Can't hurt. So here's our promoter. That's where the TATA would be. Here's where the piece that would actually be read by the ribosome. Once you make the RNA, remember this is DNA, you make the RNA, well, the RNA is going to be a copy of the piece that's in yellow here. The terminator is the thing that makes the polymerase stop copying. So this is the place the RNA polymerase attaches. Here's where it starts copying, and it stops copying when it hits a terminator. All right. So read through this, realize that a gene has several, there are many genes, I should say, on a chromosome. This is probably not an accurate representation of it, but there will be thousands of genes on each chromosome. Little places that are what? That have a promoter and a terminator. So in other words, in that long sequence, you have promoters and terminators, and anything in between is what's going to be coded for a protein, and that defines a gene, and it determines the protein that's made, and there will be thousands of these little sequences that are between promoters and terminators along here, and wherever you have one, that's going to be a place where mRNA can be made, and that can potentially go to a ribosome and be translated by the ribosome into an amino acid sequence, which will fold into a protein. All right, so just keep in mind DNA, chromosomes, nucleus, cell. Realize bacteria are much simpler. All right, and that there are mitochondria and chloroplasts that have their own DNA. All right, there is this concept called the endosymbiotic hypothesis, and I think I'll probably post that on Monday so that you can learn about that one. And let's see, I think that's it. This is actually a pretty easy one, except for maybe the number 4 here, where you have to do the hairpin loop. You need to know about them, and if you can do this, I'll be happy to give you extra credit. All right, and that's it for Worksheet 2. This is the easy one.